you very much, Lydia. Thank you, Wiseman. Thank you, Francis, for the invitation. It's a pleasure to be here. I'm probably the least informed person in the room because I'm a population geneticist, and I just happened to come across this fascinating species, and I decided to check if there was any genetic data available that allowed us to understand its diversity and population structure. And I realized that there was none. There was nothing. So until we started working on mopane worms, there was a very old study based on protein polymorphism on some enzymes. It was before the invention of DNA that it was performed. And it focused on two populations, one in the Kruger Park and one outside the park. And they found a reasonable level of genetic diversity based on those markers. And after some years of trying to secure funding and people who were interested enough, who did not see this as a very weird eccentricity from my part, it took some time for my projects on mopane worms to gain the respect of a lot of people. But I think that I managed to change that perception, and now they are seen as species that deserve to be included in these discussions, to the point that I have the pleasure of highlighting this photograph of mopane worms here in our poster. And it's a huge pleasure to see that these critters are taken more seriously than ever before. So what was shocking to me, not only the absence of genetic data, but also the contrast between the absence of genetic data and the relevance that the species has to so many people, from the nutritional aspects to the cultural aspect, to the ecological impact on the environment. So it's been a pleasure and an honor to work with these organisms. So what we did, we sampled mopane worms, this species, particularly Gonimbrasia bellina, that in its adult form is a very beautiful moth. We sampled specimens in Namibia, in Botswana, and in South Africa. So from zero genetic data, we went to a lot of genetic data, 160 individuals, 24 sampling sites. And what we discovered was that the species is phylogeographically structured. That means that there's a branch that we can say it's typically Namibian in terms of its diversity, and a branch that it's typically Limpopo River Basin. That's the area that we focused on. And what we see is that the populations seem to be divided by the border line, quite interestingly. And there's only mixing or inter-gene flow at one sampling point, where populations from South Africa and from Botswana seem to be exchanging genetic material. So we can also see from the different shades of the colors that we're showing here that Botswana has areas where there's only one type, and South Africa as well. So diversity is presently not great when we look at the mitochondrial DNA analysis. But this is only half of the picture. But it allows us already to say that lowest genetic diversity in Botswana, one contact zone, and we see that populations seem to be isolated with genetic gene flow at these sampling points in what I call the Limpopo River Basin. In terms of the nuclear genetic markers, that means what happens in the rest of the genome. These analyses were only performed for samples collected in South Africa and Botswana. And what we see is strong genetic differentiation at each sampling site, unique gene pools, low genetic diversity, alarmingly small effective population size, and signs of recent bottlenecks. So what we're seeing from the genetic data confirms the concerns that have been shared for at least 30 years, we're talking about 3 decades, that concerns have been voiced by practitioners in conservation and by other professionals involved in wildlife. They've been calling attention to this problem for at least 3 decades. And nothing has happened in the meantime, because insects in general fall through the cracks of conservation strategies. They fall through the cracks of genetic assessments. They fall through the cracks of legislation. They fall through our emotional cracks, because generally we don't want to be around insects. And they play an immensely important role in ecosystems. And species such as this one that inhabits relatively poor environments where every organic molecule counts, the impact of their disappearance should be estimated. And that's probably going to be one of the projects that I need to implement next. What would happen to mopane woodlands if we removed mopane worms? But it's much worse than that, in my perspective. What we should be thinking about is what would happen to Southern Africa if mopane woodlands are removed. We're not talking about removing one species on purpose. We're talking about erosion of huge areas that are not rainforests. They are not heavily forested as in traditional forests, but they are still woodlands and they still play a very important role. So the gut of a mopane worm is a magical system where things are created that could not be created in any other way. The gut microbiome of the mopane worm adds value to the foliage on which it feeds, and it returns it to the ecosystem in a way that plants and other organisms can utilize. So what is it that we know on top of this? Almost nothing. The ecology and biology are now starting to be addressed by researchers at Stellenbosch University. They're looking at the contribution of frass to the recycling of soil nutrients. The socio-economic aspects have been objects of studies, but things have been evolved so quickly that I think that a lot of what we thought we knew is now outdated, because harvesting used to be a household level activity that basically fed the community, and maybe a little bit of excess was sold. And now what we see is commercial level harvesting by big groups of people that are transported under the leadership of a commercial harvester. So it has become big business. Last time the volume of trade was estimated. It was estimated at $85 million US, but I think that nobody knows actually. Okay, so we lack research on everything that's related to the mopane worms, including what is it that's happening to mopane woodlands as we speak, because they are under immense pressure from increasing human populations and the needs for fuel, for wood, for housing, for everything. Also, diapause at this point does not allow for the species to be farmed. That means that there are 1 or 2 generations per year max, and we cannot continuously breed them throughout the year. Pests and diseases are also poorly understood. What is the impact of climate change? What are the dynamics of outbreaks? What determines when and where these huge numbers of mopane worms miraculously appear at the same time? And also relationships with other species are largely unknown. On my side, or my lab side, what we can do about this is to help with our expertise. We have a project that aims at completing the genome of the species, and also to analyze the pangenome and the microbiome that plays such an important role in the ecology of mopane woodlands. And that's it. Thank you very much.